So now let us start with the experiments conducted by Mendel and study about their conclusions. So first we'll come about what is called as a monohybrid cross. Now mono, the term itself indicates mono means single, one. So a monohybrid cross is a cross where the inheritance of a single character is studied. It doesn't mean that the only one character is being inherited from the parents to offsprings. There are many characters being inherited from parents to offsprings, but the observation and study will be only on one character. We are going to only observe on one character. So the cross in which inheritance of one character is studied is called as a monohybrid cross. In fact, Mendel first started his experiments on seed shape. But nevertheless, we will understand the monohybrid cross by taking the basic contrasting character of plant height, because you had already done it in class 10, and it will be a recapitulation for you and it will bring about a better understanding also for you. So let us start off how to do the cross, and before we do the cross you need to understand, since these are diploid organisms, the alleles can only express themselves when they are in pairs. The alleles cannot express themselves when they are alone. They can express themselves only when they are in the form of pairs, because it is a diploid organism. So please keep this in your mind. So what did he do? He took two parent plants and he took the pollen grains from one plant and he cross-pollinated it onto the stigma of another plant. Now what do you think must have been the techniques he must have used? He must have used the methods of, if you remember, emasculation, clipping of anthers, and then bagging, so that no other foreign pollen can come and pollinate that particular stigma. Rather, he used a desirable pollen and he put it on the stigma. So in the first chapter we had done these techniques, emasculation and bagging, wherein we talked about cross hybridization experiments, and those were the techniques taken up by him also. So he took the pollen grains from one plant and put it onto the stigma of another plant. So we are going to consider one male and one female. So what is the character he is considering? A pure tall plant crossed with a pure dwarf plant. He is considering a pure tall plant crossed with a pure dwarf plant. So this is the phenotypic expression. So what is the genotype of such a plant? Since it is a pure line, you remember homozygous condition? Since it is a pure line, this tall plant has to be capital T, and this dwarf plant of course has to be small t, there is no other option, right? Recessive alleles can express themselves only when they are in homozygous condition. So pure lines means homozygous plants were taken, tall plant and a dwarf plant. So capital T and small t, and these are called as pure lines. What are they called as? They are called as pure lines. So the genotype is this. So during sexual reproduction, what happens? Meiosis occurs. When meiosis occurs, we learnt in class 11, one set of chromosomes will go into one gamete and the other set of chromosomes will go into the other gamete. So the two alleles for a particular character get separated out in the gametes. So a gamete will have only one allele of the pair and the other gamete will have only one allele of the pair. So when two gametes fuse, these alleles again come back close to one another and they pair off. That is when the expression comes. So nevertheless, so what will be the gametes for this particular genotype? The two gametes will separate out due to meiosis. That is, one allele will go into one gamete and the other allele will go into another gamete. And then similarly, you have this also having two gametes, and each of the gamete is going to receive one allele, that is small t and small t. Now here you can see the same type of gametes or the same type of alleles in the gametes, and here also it is the same type of alleles in the gametes. So when these were crossed, this is the male and this is the female. When these gametes are crossed and fused, you can find out about the combinations by using a square called as Punnett square. There is a square called as Punnett square, which is actually named after the scientist who actually devised this for these hybridization experiments. Reginald C. Punnett, and hence the square was commonly called as Punnett square. So you just basically have to make columns based upon the number of gametes, say the male gametes and the female gametes, and then these two gametes will come here and these two gametes will come here. Then a male and a female gamete fuse with each other. So you have capital T small t. Here again you have capital T small t. Again you have capital T small t, and again you have capital T small t. So the first observation of his cross hybridization experiments between tall and dwarf plant, these were all the deductions he had done or after he had observed. He came out with these letters and this kind of calculations. But what you need to understand is initially he was only observing them. He did not do these calculations. So when he crossed a pure line tall with a pure line dwarf, he was surprised to see that all the seeds which were produced by this cross, when they were again sown in the field or the garden, they gave rise to only tall plants. All the plants were tall. And this generation, the first generation of the offsprings, was called as F1 or the first filial generation. What is it called as? F1 or the first filial generation. So he was surprised to see that even though he crossed a pure tall plant with a dwarf plant, he was surprised to see that all the seeds were growing up as only tall plants. And then he was wondering, where did the dwarf factor go? If there was a dwarf plant, why is the dwarfness not being seen in this generation or in this F1 generation? So then he decided to take two plants from the F1 and cross them again to see if they are able to express the dwarf plant or not. Right? So till here was the F1 generation, and then comes the F2 generation. So what happened in the F2 generation? I'll just take up in the next clip. So I just hope this is clear, children, what are pure lines and how the F1 generation is formed.